All right, in this video, we're going to talk more about the things that make up the cells, the organelles. So most of the organelles we're going to talk about are found in eukaryotes, but some are found in prokaryotes as well. So here we have two eukaryotic cells, the animal cell and the plant cell, and within those two cells you can see pretty much all the different kinds of organelles that are found in cells. So all cells have a cell membrane that surrounds the cell, and then the stuff inside the cell membrane that's not in the nucleus is called cytoplasm. The portion of the cell outside the nucleus that's within the cell membrane. So the nucleus is found only in eukaryotes, and it controls most of the cell processes and contains DNA. In prokaryotes, they have DNA, but it's just in an area that's not enclosed or separated from the rest of the cell, called the nucleoid region. So here you can see the nucleus. The chromatin is the DNA, the genetic material. The nucleolus is an area that is where ribosomes are being produced. And to allow materials to move in and out of the nucleus, we have pores in the nuclear membrane or the nuclear envelope called nuclear pores. So the envelope or the nuclear membrane is what separates the nucleus from the rest of the cytoplasm. So first let's talk about the organelles that build proteins. So ribosomes are small particles of RNA and protein found in the cytoplasm of both prokaryotes and eukaryotes. The structure is a little different, but they both have them and they both have the same function, and that is to produce protein. The endoplasmic reticulum, or the ER, is an internal membrane system found in eukaryotes only. And there's two types. There's rough ER, coated with or covered with ribosomes. It gives it a rough appearance, and it's involved in protein synthesis and processing. The smooth ER doesn't have any ribosomes. It's involved in lipid and carbohydrate production, as well as detoxification of drugs. The Golgi apparatus is a stack of flattened sacs that package proteins for export from the cell into vesicles. So you can see kind of the process here. So the nucleus contains the genetic material, has the instructions on how to build the protein. The ribosomes are where the proteins are made. If the ribosome's on the rough ER, it's processed there, travels to the Golgi apparatus, where it's packaged into a vesicle and heads to the cell membrane where it leaves the cell. Here's just an overview of the process. So there are different structures within the cell that are membrane-bound sacs that have various purposes: vacuoles, vesicles, and lysosomes. So vacuoles are large sac-like membrane enclosed structures that store materials like water, salt, proteins, and carbohydrates. Some of the freshwater protists have central vacuoles that collect excess water and expel it from the cell, but mostly they're for storage. Vesicles store and move material between cell organelles as well as to and from the cell membrane. Like we talked about earlier, the proteins that were packaged in the Golgi apparatus travel to the cell membrane. Lysosomes are organelles filled with digestive enzymes that help cells break food and things down into usable bits of matter. You can see there's a contractile vacuole in the paramecium, the central vacuole of a plant, and so on. The cytoskeleton is like the framework for a cell. It's made up of various protein filaments that give cells their shape and some internal organization and help with transportation through the cell. There are different kinds of protein filaments. Microfilaments are one type. They're made up of a protein called actin. They help support the cell and they help the cell move. Then are the microtubules, hollow structures made of another protein called tubulin. They make up structures called centrioles that are only found in animal cells that form part of the spindle apparatus. We'll talk more about that when we talk about cell division later on. They also help build the various projections from the cell surface, which help with movement of materials across the surface as well as moving the cell, the cilia and flagella. Chloroplasts are only found in cells that do photosynthesis. So we're talking about things like algae and plant cells here. Chloroplasts capture energy from sunlight and convert it into food that contains chemical energy through the process of photosynthesis. Mitochondria also convert chemical energy, but in this case they take chemical energy from food and we break that food down into components that cells can use. The process producing ATP, which is a form of energy cells can use for them to do work. Both plants and animals have mitochondria. The process that happens in the mitochondria is called cellular respiration. Now, both of these organelles are different than other organelles in that they have their own DNA, which is different from the DNA in the nucleus. It's similar to bacterial DNA more so than it is to eukaryotic DNA. So some cells have a cell wall, a strong supportive layer that lies just outside the cell membrane. This is bacteria, fungi, and plants that have cell walls. Everything else has a cell membrane but no cell wall. The cell membrane is what regulates what gets into and out of the cell by being selectively permeable. Some things can go through it, some things cannot. Like I said, all cells have a cell membrane. Here you can see the cell membrane and the cell wall of the bacteria cell at the bottom and the plant cell on the right. So the cell membrane is a phospholipid bilayer primarily with embedded proteins. And it again separates the inside of the cell, the cytoplasm, from the outside of the cell. The phospholipids have an area that's hydrophilic or water-loving called the head, and a hydrophobic tail that doesn't like water, water-fearing. So you can see the arrangement is so the heads face the outside and the tails are on the inside. There's proteins that allow things to move in and out of the cell, help with communication and identification, and so on. So the model that explains the function and the structure of the cell membrane is called the fluid mosaic model, because the proteins can move throughout. They're not fixed in place. They can flip around. They can move around through the phospholipid bilayer.